Hello and welcome to today's webinar on cell counting technologies from Chemometic. We're very pleased to have Tanya Seeger with us today to discuss the role of cell counting in cell viability testing using their technology platform. And as a special note, we were very pleased that Chemometic was one of the first sponsors of the first Boston BioLife meeting on degenerative disc disease using stem cells and biologics. We still have Chemometic as a partner, and we're moving forward now with our online education. So Tanya, thank you so much for being here today, and we're really looking forward to hearing about the important work you're doing in cell counting and cellular viability studies.
Welcome everybody to today's webinar about counting mesenchymal stem cell for cell therapeutic applications. My name is Tanya Seeger and I'm a field application scientist at Chemometic, which is a company specialized in cell counting. As you may all know, mesenchymal stromal cells are multipotent stromal cells which can be used for cell therapeutic applications. Those cells can be easily isolated from various sources. So most common are adipose tissue and, for example, bone marrow as well. But why are MSCs so interesting for regenerative therapies? First of all, MSCs can be easily injected systemically into the body, and those cells are then mobilized to the damaged tissue and secrete bioactive molecules to support the healing process. Another important point is that MSCs can be differentiated into bone, fat, cartilage, and muscle in vitro and in vivo. And this is very interesting for regenerative therapies, which are aiming to replace damaged cells and tissues. Furthermore, MSCs can modulate the immune system, which is interesting for the treatment of inflammatory diseases or in combination with hematopoietic stem cell transplantation to suppress the host immune system. Until now, around 600 clinical trials are running at the moment for the treatment of different diseases. Here, the major part is focused on the treatment of bone diseases, cardiac diseases, musculoskeletal disorders, and disorders of the central nervous system. When we are talking about cell therapies, we should distinguish between allogenic and autologous therapies. An allogenic therapy is a cell therapy where the donor is different from the host. Manufacturing of such therapies are much cheaper than autologous therapies. In comparison to that, an autologous therapy is a cell therapy where the patient's own cells are used to generate the therapeutic cells. These cells don't give rise to a response from the immune system. MSCs can be used for cell therapeutic applications, and those cells can be isolated from different sources. As I've already mentioned, for example, most common are cells from adipose tissue and from bone marrow. Adipose-derived MSCs are highly proliferative and can be easily isolated from the so-called stromal vascular fraction after biopsies and liposuctions. For cell therapeutic applications, the stromal vascular fraction can be directly used for patient treatment. For isolation of bone marrow-derived MSCs, bone marrow biopsies or aspirates are taken. The bone marrow aspirate is purified using fecal gradient centrifugation, and the bone marrow-derived MSCs can be stored and expanded before re-injecting back into the patient. During the whole manufacturing process of adipose-derived MSCs and bone marrow-derived MSCs, it is very important to determine the cell numbers. Here, for example, the evaluation of the cell dosing, the so-called cell count, and the sample quality, the so-called viability, is very important for improvement of the patient treatment. If you would like to count your cells, you should do it in a precise and accurate way. Here, precision is the closeness of two or more measurements to each other. Precision is quite important because of reproducibility and reliability of your counting results. In comparison to that, the accuracy is the closeness of a measured value to a standard or known value. But accuracy is quite challenging to address in cell counting, as there is no real reference method until now. So counting, for summing up, should be accurate and precise. But how to get the most accurate and precise cell count? So to date, there are different methods available. For example, you can detect the cell count and the viability of your cell sample using, for example, bright field and trypan blue. Here, using the trypan blue exclusion method, viable cells with an intact cell membrane will not be stained with trypan blue solution in comparison to dead cells with a permeabilized cell membrane. This you can do manually by just using an hemocytometer, or you even can use automated cell counters which are also using the trypan blue exclusion method. Another idea is to use the Coulter principle. This is using electrical impedance, meaning particles and cells are suspended in electrolyte buffer and are aspirated through a pore inside a measuring capillary in a low voltage electric field between two electrodes. Each particle will cause an electrical pulse which is recorded and later on translated into the total cell count and the viability. You can also use flow cytometers for detecting cell count and cell viability of your cell sample. Most of the time here the sideward scatter and the forward scatter is used to detect the granularity and the cell size and later on to detect the cell count and the cell viability. Another way or method to detect the cell count and cell viability is to use fluorophores and microscopes. For example, using of DNA fluorophores. In our case, our NC200 instrument, this is an image cytometer which is using acrid in orange and DAPI for determining cell count and cell viability. Our instrument is using, for example, acrid in orange and DAPI. Acrid in orange is staining all cells. DAPI will only stain the dead cells. Those two fluorophores are already lyophilized inside of our disposable VIA1 cassette. So in the end, the only thing what you have to do is you have to insert the VIA1 cassette into your cell suspension. Just press the little piston on top of the VIA1 cassette. The cell suspension will be soaked inside, mixed together with acrid in orange and DAPI for staining all cells and only the dead cells, and later on be analyzed in the analyzing window in the VIA1 cassette. This will be recorded using our image cytometer NC200 and later on translated in the software in the viability and this total cell count. This will give you a very consistent cell counting because our instrument and the VIA1 cassette is already pre-calibrated, which will eliminate every human bias. But what is so special about the NC200 instrument? It is so special because we are using DNA fluorophores. For example, the really common DAPI fluorophore, which can clearly discriminate cells from other particles. Just remember, you would like to detect the cell count and the cell viability in your bone marrow derived and adipose derived cell suspension. This cell suspension not only consists out of your stem cells, there are many other things inside as well. For example, cell fragments, fat droplets, microvesicles, and erythrocytes. As the DAPI fluorophore will only bind to DNA, this is really specific in cell counting. For example, when you are using trypan blue and a bright field microscope to detect your cells in your cell suspension, you will maybe get such kind of image. In here, it is very challenging to distinguish between cells and cell fragments, and even to detect if the cells are viable or not. In comparison to that, when you're using DNA fluorophores, it is much more clear what is a cell and what is just cell fragments, because now you can really distinguish between cells and cell fragments. Now let's talk about the pitfalls when counting bone marrow-derived MSCs. For isolation of bone marrow-derived MSCs, bone marrow biopsies or the aspirates are taken. The determination of cell count and viability directly from bone marrow samples is especially challenging, as these contain great levels of erythrocytes, which interfere with conventional automated and manual counting methods. For easy and reliable counting of MSCs directly in bone marrow aspirates, the nucleocounter instruments offer a fast protocol to cope with the erythrocytes in the sample using our solution 17 erythrocyte lysis buffer. By adding a blood lysis buffer to the diluted bone marrow aspirate, the erythrocytes will be lysed and the hemoglobin diluted. Afterwards, the sample is stained with the two fluorescent dyes, acridine orange and DAPI. And in the end, the viability and the total cell count can be easily recorded. If you would like to determine the cell count and the viability of your adipose-derived MSCs, there are other pitfalls. The stromal vascular fraction contains a bunch of different cells, so MSCs, stromal cells, endothelial cells, adipocytes, erythrocytes, and as well many lipid droplets and micelles. Many counting techniques fail to discriminate between cells and artifacts, or will give high variations between different users. To overcome this problem, the nucleocounter instruments offer an effective protocol to determine cell count and viability of adipose-derived MSCs using our solution 10 lysis buffer. I would like to show you a small movie on how to use the solution 10 assay.
The nucleocounter NC200 offers automated cell counting, optimized for several different cell types, including stem cells and adipose-derived mesenchymal stem cells, often used in regenerative medicine. With the use of our innovative lysis buffer, we can, if needed, separate clumps of cells and remove adipose-derived micelles from the sample. The volume-calibrated VIA1 cassette contains DAPI for cell staining. First, insert a VIA1 cassette with a sample of cells mixed with the lysis buffer for the total cell count, followed by a VIA1 cassette with a sample of cells without the lysis buffer for the non-viable count. In less than 1 minute, you have your results on screen, in your laboratory management system, or as a PDF to save or print. The NC200 is 21 CFR Part 11 ready, and with the ability to perform IQ, OQ, PQ, it is ideal for regulated environments. So if you need a powerful, reliable, and consistent cell counter for your stem cell needs, look no further than the nucleocounter NC200 from Chemometic.
If you would like to count your stromal vascular fraction cells, you need to do two measurements. First one would be take out some small amount of your cell sample into a new Eppendorf tube and mix it together with our solution 10 lysis buffer. Solution 10 will lyse all cells and only leave the cell nuclei available for staining together with DAPI, which is already included in our VIA1 cassette. So meaning after lysing all cells with solution 10, all of your cells can be stained with DAPI, and this will result in the total cell count. If you are also interested in the viability of your cell sample, then you need to do a second measurement. So again, take out some small amount of your cell sample and only insert it via one cassette. Now, only the dead cells will be stained with DAPI, and this will result in the dead cell count. Those two measurements will be combined in our instrument to the total cell count, viability, and the live cell count. The NC200 is routinely used for counting stromal vascular fraction cells together with the Q-Craft instrument from Carestream America. The Q-Craft can be directly used during a surgery, and afterwards the cells can be collected from the stromal vascular fraction and again counted using our instrument. Let's talk about the results that you will get out of the NC200. You will get a fast and automatic determination of cell count and viability. And you will also have the possibility to further evaluate and verify the results using our unique image overlay function, in which you can include and exclude cell fragments. For example, in here you will always get some scatter plots in the raw data files. In here we can see the DAPI total intensity displayed on the x-axis and the DAPI total area displayed on the y-axis. Everything what is in the blue box in the scatter plot is your cell count, and this can also be verified by checking the results on the upper left side. In the fluorescent image, all cells are displayed. Those cells can be easily distinguished from cell fragments, which are, for example, visible in the red box. And also here again, we can verify the results in the lower left red box, where one cell is just displayed. Those protocols can be adapted and saved for further usage. Our instruments are very precise and reproducible. This is due to our Via 1 cassette. The Via 1 cassette, our consumable, is calibrated, meaning you will have always a very consistent cell sampling and workflow. The calibrated volume eliminates all production variation, and this you can see later on in linearization of dilution ranges with a very low CV value. Together with our calibrated instruments, this can very much ensure consistency in your cell counting, meaning it doesn't matter when you've ever bought an NC200 instrument or where you've bought it, they will completely count the same. Meaning together the calibrated Via 1 cassette and the calibrated instrument will help you to reach a very high precision and reproducibility in your cell counting results. Our NC200 instrument can be easily used in regulated environments. The NC200 is 21 CFR Part 11 ready and can be used easily for GMP environments. Therefore, we are offering electronic signatures, adapted protocols, our calibrated instrument, and our calibrated Via 1 cassette. Furthermore, we are offering installation qualification, operation qualification, performance qualification, and as well automated PDF reports and signed approvals. For summing this up, I would like to tell you that MSCs are very interesting candidates for regenerative therapies. During the isolation process, it is very important that you accurately determine the cell count and the viability. Many other counting techniques fail to discriminate between cells and artifacts, or even will give high variations between different users. But with our lysis buffer systems, we can even easily count primary bone marrow aspirates and lipoaspirates to cope with erythrocytes, to cope with other artifacts which are also inside of your cell sample. The NC200 is very user-friendly and consistent, and you can easily implement those instruments in clean rooms. Thanks the audience for your attention. I'm now very happy to answer any of your questions. Thank you.
Thank you, Tanya. That was a very informative presentation, and thanks, Chemomedic, for your continued support of the Boston BioLife meetings. As I said in my introduction, cell counting and cell viability analysis is a very important component. And what I really liked about the presentation that you did was you compared your solution to others in a non-biased way. And I think from a physician standpoint or a healthcare provider standpoint, looking at the sophistication of laboratory equipment is very powerful. Although it's not common for them to need it or use it, it certainly lends a lot of credence to what they're doing. Every day we hear about the need for standards and viability, and Chemomedic is really a leader in this effort. And so we are very pleased to have you as a partner and a sponsor in our meetings. Tanya, thank you so much for your participation today and giving us such a wonderful insight into cell counting.